Thank you, Dr. Damayanti from Dr. Stoma Hospital.
Hi, everybody. Thank you, Dr. Heri Suroto and all senior here, Professor Kaplan.
Hi.
Nice to meet you.
Professor Aziz Natter, thank you very much for joining us. Okay. Yeah.
Okay. Sorry. Okay, thank you. Welcome. It's my great pleasure to welcome you all to the 8th Erlangga Webinar Conference series as a pre-Congress of Asia Pacific Association of Surgical Tissue Bank 2021. Thank you all guests for being a part of this insightful webinar. Firstly, I want to introduce myself. I'm Molly Edward I'm an orthopedic surgeon. I will become the host here. And as you have been aware that today webinar topic will be medical signaling stem cell from bench to bedside. And allow me to introduce moderator and speaker of this webinar. There will be Dr. Heri Suroto, PhD, as a moderator. He is a Director of Dr. Stomo Cell and Tissue Bank, Dr. Stomo General Academic Hospital, Surabaya, Indonesia. And there are three inspiring speakers from this webinar. Firstly, Professor Kaplan from Case West Reserve University, Cleveland, Ohio, United States of America, who will discuss medical signaling stem cell from bench to bedside. The second speaker, Dr. Ferdian Shah, PhD, as a chairman of regenerative medicine at Dr. Stomo General Academic Hospital. And the third will be Mrs. Rizka Andalusia, as Drug Registration Acting Director, Drug Production Control, Indonesian FDA, who will discuss stem cell regenerative medicine, regulation and supporting. And I want to introduce the panelists of this webinar, Professor Dikora November Utomo from Indonesian Tissue Bank Association, or Perbaji, Mr. Bintang Sucahya, PhD from Indonesian Association of Tissue Engineering and Cell Therapy, Reja Selindo, and third panelist, Mr. Rahayu Salim, PhD from Indonesian Stem Cell Association, ASPI. I sincerely hope you will enjoy this fruitful webinar. Thank you for your appreciation. And I would like to pass this opportunity to the moderator. Please welcome Dr. Heri Suroto.
Thank you, Dr. Molly. So it is a great chance for us conducting this webinar. There are 3 speakers, and also there are 3 panelists, and also a foreign guest owner, Professor Aziz Natter, and also there is from Indonesian FDA, Dr. Rizka Andalusia. So hopefully this webinar will have a great chance for us to learn and to share each other and to discuss. Yeah. So can the slide please? Yes, thank you. Yeah. I just want to memorize the third speaker. So I will start with my PowerPoint. This is 8th Erlangga Webinar Conference Series as a pre-Congress 18 APASTAP 2021. Yeah. Proudly medicinal signaling send from bench to bedside. And this is the flyer. Okay, next. Next, please. Yes, this is just to memorize that 2004, 16 years ago, I went to Case Western Reserve University, and I hearing your experience about the research of mesenchymal stem cell at the time, and hope a fortunate 16 years later in our hospital we have a cell and tissue bank installation. So it is a great chance in this meeting we can discuss each other. Okay, next please. And all of us know about Professor Arnold E. Kaplan. Yeah, he is professor at Case Western Reserve University, Skeletal Research Center. Yeah, he is so many publication and also citation and also the high index. Okay, we can see on the screen. So this afternoon we can discuss about we can hearing from Professor Kaplan with medicinal signaling cell. Okay, next please. The next speaker is Dr. Ferdiansah. Also, the subject area, he have so many research, documentation, citation, and high index. He is a chairman of regenerative medicine at Dr. Stomo General Academic Hospital. He will speak about the development of regenerative medicine in Indonesia. Then the last speaker, okay, she is Dr. Rizka Andalusia. She is Director of Drug Registration, Acting Director of Drug Production Control Indonesia, FDA. Yeah. He also have many research and citation and also high index. Okay, so please, Professor Kaplan, the time is yours, so we can hear from your experience and research. Thank you very much. Please, Professor Kaplan.
Well, good morning, everybody. I'm happy to be here, and I'm going to talk about MSCs, and I'm going to make the very strong point that in your body, these cells are not stem cells, and I'll provide you with data for this. Okay. So this is my disclosure document, which says that I started a company called Osiris Therapeutics. I have no association with Osiris. They get some royalties, which the university splits with me very generously. And that's the only economic aspect that I need to discuss. So on June 18th, I gave a lecture about COVID-19 and MSCs. And in this lecture, I provided data to indicate that allogeneic MSCs can be given to very severely sick patients with very positive results. And so the hypothesis is that MSCs could actually be curative for this virus. And I provided some preliminary data to indicate that MSCs make molecules that are antiviral, that MSCs can calm the cytokine storm, and MSCs actually can affect tissue regeneration. I'm going to provide more data for that in a few minutes. So in 1991, I coined the term mesenchymal stem cells. And I did this because in vitro, I could make these cells differentiate into cartilage or fat or bone or muscle or a variety of mesenchymal phenotypes. So in culture, in cell culture, I could justify the term stem cells. In 2010, I changed the name because in your body, these cells are not stem cells. And so I now call them medicinal signaling cells because they're drugstores. They make drugs at sites of injury. And I'll show you the data for this. So I'm going to refer to them as MSCs. And again, I know it's a very popular name, stem cells, but again, it gives the wrong impression to patients and the wrong impression to doctors. And I'll address this issue with publications. So here's the conclusion, simple conclusion to my lecture, that MSCs come from perivascular cells at sites of injury. MSCs do not arise from stroma. Stroma is an old anatomical term that means connective tissue. MSCs do not come from connective tissue, and I'll stress that in a few minutes. MSCs do not differentiate into mesenchymal tissues. They make drugs. They make therapeutic molecules at sites of injury, and I'll give you examples of this. MSCs can manage the local immune microenvironment. MSCs can make molecules to help injured tissues regenerate. They regenerate themselves. And again, I'll discuss that in detail, and completely unexpected in the early days. And now I'll show you the data. MSCs can make molecules that sit on opioid receptors and therefore can be used to manage pain. And also they make proteins which are both antibiotic and antiviral. So this is the theme of my entire lecture. It's a very simple logic which covers almost every tissue of your body, and that is that you have the capacity, innate capacity, to regenerate every single tissue of your body. And this is the example I always use, which is every single second, 15 million of your blood cells drop dead and are perfectly replaced without you ever knowing it. So while I said that sentence, 200 or 300 million cells drop dead in your body and were perfectly replaced. These cells in your bloodstream come from this real stem cell, this hematopoietic stem cell, which is in your bone marrow. If you do chemotherapy and radiation and you lose this cell, if you don't replace it, you will die, because all of those blood cells have half-lives and they will expire on time. Also in bone marrow, there's another cell, which again I called a mesenchymal stem cell. And in the late 1980s, I drew this slide as a hypothesis slide. And in the late 1980s, it was the dogma of the day that there was only one stem cell left in your body, and that stem cell was the hematopoietic stem cell. And so when I drew this diagram and said that there was this mesenchymal stem cell that I could make in culture go to bone or cartilage or muscle or tendon, ligament, or fat, this was not well accepted. It turns out that if you want to do tissue engineering, you have to be able to manage these lineages. So for tissue engineering of cartilage, for example, you have to be able to drive these cells down the appropriate lineage, because ear cartilage, nose cartilage, throat cartilage, knee cartilage, hip cartilage, ankle cartilage are all different. And so you can't just take an MSC and simply put it down this pathway. It has to be managed every single step in this pathway to give you the proper cartilage cell or muscle cell or whatever cell you want in vitro has to be managed. But if you want to talk about regenerative medicine, you only need to talk about the MSC at the top, and that's what I'm going to focus on. I'm not going to focus on this tissue engineering aspect. So I started working, and all my early publications were on MSCs that arose, that I was able to isolate and characterize and expand in culture from bone marrow. But now almost every tissue of your body, there's a publication which says you can isolate an MSC and you can take those MSCs and get them to differentiate into bone or fat or cartilage. And so for someone like myself that works with this special tissue bone marrow, it was rather appalling, for example, to know that you could get MSCs from fat. I can't think of anything more disgusting for someone who works on marrow to even think that fat could be a regenerative tissue. But indeed, you can get MSCs from fat. And what's missing from this slide is the fact that all of these tissues have blood vessels. And that's the connector between MSCs and the appearance of these cells in your body. So I'm going to give you the conclusion first, which is that all MSCs come from perivascular cells. So this is a capillary in heart, and these red cells that are grasping the capillary, they're the progenitors of MSCs. They're perivascular cells or pericytes. If you break the blood vessel or you inflame it, these cells come off and they differentiate into MSCs. And that's the origin of MSCs. And if you isolate pericytes, the cell surface marker most associated with pericytes is CD146, but also smooth muscle actin. You can see that you get a homogeneous curve. All of the markers for hemopoietic stem cells are negative in these cells, and all of these markers that we identified for MSCs, all of these are positive. So pericytes, and this paper documents that pericytes and MSCs are identical, and this is the data to show that these two are identical. And I'm going to talk at the end of this lecture about this homogeneous distribution that we see on cell sorts, because we were led astray by this, and we originally said that this was a completely homogeneous population. That's not the case. So every cell in this slide, every cell that you see on these blood vessels is a pericyte. Every cell you see on these blood vessels are pericytes. Every cell you see on these blood vessels are pericytes. You have 100,000 miles of blood vessels in your body. Every single blood vessel has mesenchymal pericytes on the outside. There are no exceptions to this. And what's of interest here is all of these pictures look different. This pericyte and this pericyte look different because they're from different tissues. And so although the pericytes give rise to MSCs, the MSCs have different chemistries because they come from different tissues. So what's clear is I know how to take pericytes off of blood vessels. I know how to put them in culture, and I know how to get these phenotypes in vitro, and that's why I called it a stem cell. But these cells are not multipotent in your body, and here's a publication that you can read in detail, and what they show is they can take labeled perivascular cells and get cartilage and bone and fat. But in this case, the labeled pericyte here in red, the labeled pericyte in animals that are aging or on high-fat diet, or you break bones or you injure them, the pericyte never is found, the label is never found in any of these newly differentiated tissues. And so this paper documents that the pericyte is not the progenitor for these different mesenchymal tissues. This is another publication, and this is a very important publication because in this case, it says that MSCs don't stick around for a very long time. they indeed get consumed by monocytes or other phagocytic cells, and they're gone. So in this case, where you take and put MSCs into an interarticularly, and you see cartilage regeneration, that MSC that goes into that osteoarthritic knee does not differentiate into chondrocytes, and I'll come back to that in the next slide. So there's no evidence that the MSCs engraft and differentiate. As a matter of fact, they're cleared rather rapidly, and I'll come back to that, because that's the missing clue to how allogeneic MSCs can be used for most medical uses. So again, I want to come back to all of these tissues, because we missed something fundamentally important in all of these tissues. And I summarize it very simply here, and that every single one of these tissues has its own unique stem cell that I color here in yellow. That stem cell sits on a MSC pericyte, and both of these cells are in contact with the blood vessel, with the endothelial cells. And this is indeed the universal tissue-specific stem cell niche. So this is, you have a cardiac stem cell, you have a liver stem cell, you have a pancreas stem cell, you have a kidney stem cell. All of these cells have neurostem cells. All of these cells, these individual tissue-specific progenitors, are sitting with a pericyte next to it. The pericyte is giving it certain molecules, and both cells are sitting on a blood vessel, and that's the progenitor cell niche in every tissue of your body, and I'll come back to that. So you get an injury, the pericyte comes off. So now the pericyte is in an injured microenvironment, and it differentiates now into a new cell. It differentiates into an MSC. That MSC is activated by the microenvironment that it sees, and so it's all local. So from the front of the MSC, it makes a curtain of molecules, that stop your over-aggressive immune system from looking at the injured tissue behind the MSC. This is your first line of defense against the start of autoimmune diseases or autoimmune reactions. So, and we know the molecules made by the MSCs, and these molecules made by the MSCs that make this curtain allow us to use allogeneic cells, because the curtain actually allows the MSC to evade interrogation by your immune system. So someone else's MSCs in my body would not immediately be seen by my immune system. From the back of the MSC, it makes molecules that I call trophic, which are anti-scarring, manage pain, etc. And this sets up a regenerative microenvironment. And so the MSCs, they function as local mediators of tissue regeneration and of immune modulation. This is hugely important. And this is how these cells are used now therapeutically. So if you go to this website, clinicaltrials.gov, and you put mesenchymal stem cells in the search engine, today there are 1,137 clinical trials listed on this website. Here's the world map of all of the distribution of all of those. And this is a map of the United States. I'm politically very active. I do not like red states in our red-blue configuration, but I love red states here because it's the most highest number of clinical trials using MSCs. And here in the north of the United States, in Minnesota, there's a huge number of clinical trials, quite different than any of its neighbors. And this is because Mayo Clinic is here and has a whole program with MSCs. So actually there's more clinical trials with MSCs in Minnesota than in California. So it's quite interesting aspect. And I blew up this map because I'm just telling you in Indonesia and in Asia in general, you have to increase your number of registered clinical trials with MSCs because we want to see all of this data that you gentlemen and ladies have been talking about. So I copy pasted all of the clinical symptoms that are found on this website. And I put these in higher font for those of you who are old like me and need higher font sizes. And what you see are Crohn's disease or kidney transplantation or rheumatoid arthritis or acute myocardial infarct. All of these clinical symptoms can be affected by exposure to MSCs. And the difference between me and a 5-year-old is vascular density. So I am an old man, so I have very low vascular density. So I need a booster shot. I need allogeneic MSCs to help me with my acute myocardial infarct. A 5-year-old breaks a leg, 3 weeks later, the bone is healed, regenerated. I break a leg, it's 3 months or more. So again, MSCs are not stem cells. They're cells that make drugs at sites of injury. And so I've changed the name to medicinal signaling cells. You can call them whatever you want. You should continue to call them MSCs because I have a very delicate ego. I prefer that you do that. But I'll give you a choice. You call them MSCs or call them Kaplan cells. The problem with that, I can't get any of my papers published when I call them Kaplan cells. So MSCs that are put into the bloodstream dock at sites of broken or inflamed blood vessels because that's where they do business. They have an immunocompetent component and a regenerative component, and they are the site-specific managers of your innate capacity to regenerate tissues. And what's missed by many people is that the MSCs that dock at different sites are sensory. They survey that site, no matter where it is in the body, whether it's the liver, the heart, or stroke for brain, and they have different response profiles. So this is a very interesting publication. They take MSCs, these blue cells, and they expose them to inflammatory microenvironment, and they make anti-inflammatory molecules. But if you take the same cell and you expose it to bacteria, they make inflammatory molecules, because if you have a bacteria, you want the MSCs not to inhibit the cells from the immune system from coming in. You want many of them to come in and take care of those bacteria. Very important aspect. And again, I'm going to talk about the management of pain. And in this case, in rodents and rats, they cause the sciatic nerve injury, very painful, and they completely eliminate that injury, that painfulness, by infusing MSCs into the animal. And what's important about this paper is that bone marrow MSCs and fat-derived MSCs were equally potent in reducing the neuropathic pain in these rats. So again, my thesis is an MSC is an MSC is an MSC. The MSCs have different modes of functioning, But in the case of pain, whether it's from fat or from umbilical cord or from marrow, they're equally potent. So all of us bump into a wall and we hurt our arm and it hurts like hell for about 20 minutes. And the next day it's black and blue. So we know that blood vessels have broken here. And what happens is that you get the release of MSCs. They see the screaming nerve and they make molecules that sit on the opioid receptors. Nothing's regenerated, nothing is fixed at that point. But what is fixed is the nerve stops hurting, and that's what we see. This is Dr. Chang, one of esteemed colleagues at the Cleveland Clinic, and in 2015, he put this abstract in the annual meeting of the American Academy of Pain Medicine, and it describes fabulous experiments that were done in rats in his laboratory. All of us become tolerized when we take opioids. So for humans, you get somewhere around 10 or 16 weeks. After that, you become tolerized. And so in order to get rid of the pain, we increase the dose, and that's the basis for lots of this opioid problems that we're now having. What Dr. Chang did is he tolerized rats to opioids. He then transplanted human MSCs, human MSCs into these rats, and they lose tolerance. And the better experiment, the one I like best, is he gave the MSCs first, and then he tried to tolerize these animals, couldn't do it. So this is actually the first real data that MSCs can affect opioid-induced hyperalgesia, and also that they make molecules that sit on opioid receptors. And Dr. Chang and his colleagues published this article. You can go read this article. And this is a clinical trial that's been described in the Journal of Pain Research. 10 female patients with horrible trigeminal nerve pain. These patients had liposuction, and so they gave fat, and from that fat they isolated what's called the stromal vascular fraction, and the stromal vascular fraction, by sorts, about 40 or 45% of those cells were MSCs. There were some endothelial cells, and there are some monocytes. They injected these directly into the trigeminal nerve area, and then they did follow up 1 week, 1 month, 3 months, 6 months, and did pain scores. And the pain scores went from 9, which meant that these women were horribly into pain, and the pain scores went from 9 to 1 in 7 out of 9 of these patients, and also many of the patients stopped taking gabapentin, which they were using for pain management. This is a very important paper, and I have to say in 2011, I missed this paper, but this paper gives you another piece of hugely important information. So these are rats, which they did a trigeminal nerve pain. And so if you touch the cheek of the rat jumped. And if you give them opioids, they don't jump when you touch their cheek. Or if you give them human MSCs, bone marrow MSCs, they don't jump. And so every week they would take this molecule, which is a peripherally acting opioid receptor antagonist. So it knocks the molecule off the opioid receptor. And for the first 3 weeks, actually, this antagonist knocks the molecule off the receptor. So if you touch the cheek of the animal, they jump. In this case, if you do it on week 5, it doesn't work. And what's happened is that the pain management has shifted to the back of your head, to the brain, to the central nervous system, and you need a different kind of molecule, an RNA inhibitory molecule, which works on mu receptors. So this is hugely important. It says the bone marrow MSCs on mechanically hypersensitive areas put out molecules that first sit on peripheral opioid receptors, and those molecules can be knocked off with antagonists. And then something happens where the brainstem and the descending pathways of the central nervous system take over by week 5. So I can assure you that the MSCs that are put into these animals are long gone by week 5. So the question is, what cell is crossing the blood-brain barrier? What cell is now affecting the mu receptors? This is hugely important point. So I want to summarize all of this by saying MSCs affect pain by their paracrine capacities. The MSCs are effective for both central and peripheral nervous system. They of course reduce inflammation, which is a part of pain management, but the regenerative activity and the fact that these MSCs make molecules that sit on opioid receptors, hugely important aspect of their functionality. And again, these MSCs are functioning locally. So again, this gives me the justification again for calling these cells medicinal signaling cells. So there are many, many companies that are producing MSCs. Again, remember, I started this company, Osiris. Osiris was purchased by Mesoblast several years ago, and Mesoblast has now actually completed a phase 3 trial for degenerative disc disease and low back pain. And this is a spectacular use of MSCs. So you have an MRI hot disc, and under fluoroscopy with a long needle, they deliver 6 million total, 6 million allogeneic culture expanded marrow MSCs to this site. And they've reported publicly 100 patient phase 2 clinical trial, the results of which you can read on the Mesoblast website. And the 3-year outcome data